Hello everyone, welcome to the chapter, the disorders of immune system. So this is given as chapter 6 in your Robbins. So there is a reason why I am going to start this chapter along with carrying out the sessions on other chapters, because it is one of the most extensive chapter in pathology. Disorders of immune system, neoplasia. Fine, these are two of the some 4 to 6 chapters in Robbins, which are very detailed, very vast, very extensive, that we require proper basic or proper continuous reading in order to understand the concepts properly. That is the reason why I have chosen this chapter to start now, because this chapter will be useful when you start studying immunology in microbiology. Fine. So now let us see the normal immune response. You know, immunity means it is the protection from infectious pathogens. We have innate immunity, adaptive immunity. Innate immunity is called as the natural, it is the intrinsic mechanism of the body to react immediately as a first line of defense. Adaptive immunity, they are acquired or a specific immunity, fine, whereas the innate immunity is not specific. Adaptive immunity means the immunity we acquire as a result of exposure to foreign body antigens. Now let us see some features of this innate immunity. What are the components, features of acquired immunity and its components? Innate immunity, it is the first line of defense. Suppose you are getting an infection. In the initial 0 to 12 hours of infection, what you are getting is nothing else but your innate immunity. Let us see what are the components of this innate immunity. This picture explains the components of innate immunity. Number 1, epithelial barriers. Number 2, phagocytes. Number 3, complement cascade. Number 4, dendritic cell. Number 5, NK cell. Straight away MCQ question. All of the following are components of innate immunity except. This will be the MCQ. Once this is done, now let us see. Okay, these are the components I have told you. Neutrophils, macrophages, dendritic cells and NK cells. And we have something what we call as ILC. What is ILC? Innate lymphoid cell. Actually the natural killer cell, they are a type of the ILCs. Okay, I will explain that very soon. So we have lot of barriers. Out of that, epithelium acts as one of the mechanical barrier. The epithelial cells have it produces some antimicrobial substance like defensins. These defensins will prevent the microbes from penetrating the endothelial boundaries. Then we have monocytes and neutrophils. They are the phagocytes. Already we have discussed in inflammation chapter. Some tissue resident macrophages. We have something what we call it as Kupffer cells. Kupffer cells are there in liver. We have microglial cells, alveolar macrophages. Am I right? Yes? Okay. So these are the various macrophages. They are the tissue resident macrophages. Am I right? Yes. Monocytes means they are in the blood, whereas macrophages means they are in the tissues. Okay. Then we have something what we call as dendritic cells. Of course, we will discuss dendritic cells in detail about in the coming classes. But as of now, you remember that they are also a component of the innate immunity. Fine, they are present in the epithelium. They are present in the lymphoid organs and in most of the tissues. They are called as APCs. They are antigen presenting cells. So they are the one which will present the antigen to the B or T lymphocytes. Fine, that is what we call it as antigen presenting cells. We have lot of antigen presenting cells. We have the professional antigen presenting cells. We have the non-professional antigen presenting cells. All those we will come in read in detail in the coming sessions. Then we have something what we call as ILCs or the innate lymphoid cells. They are nothing else but the lymphocytes which are residing on the tissues. What is the importance of these ILCs? See, this is a very important potential question. The ILCs lack T cell antigen receptors. That means they don't respond to antigen. Since they don't have what is T cell receptor, antigen receptor, TCR, I will tell it very soon. So it is this receptor which helps them to bind with the T lymphocyte. Since the innate lymphoid cells, they won't have this receptor, they can't present antigens, they can't bind with the T lymphocytes, okay, all those, okay, yes, so they don't respond to antigens. The best examples of these innate lymphoid cells are what we call it as our NK cells or natural killer cells. Okay, they are the one type of innate lymphoid cells, fine, and they mainly give protection. Of course, remember natural killer cells, B lymphocyte, T lymphocytes are a part of adaptive immunity, whereas the natural killer cells or what we call it as the NK cells, they are a part of our innate immunity. Okay, innate immunity, fine, yes. So they are a type of innate lymphoid cells. Then we have the plasma proteins. I told you, complement cascade, mannose, binding lectin, C reactive proteins. All this already we have discussed. All this already we have discussed in our chapter. What inflammation? Fine. So now we see what are the components of innate immunity. See, now let us see what are the components of adaptive immunity. We have mainly B lymphocytes and T lymphocytes. B lymphocytes mainly they will what transforms them to form plasma cells. The plasma cells will secrete antibodies. T lymphocytes, okay, they mainly present antigens to the dendritic cells. The dendritic cells present antigen to these T lymphocytes, and they are also called as effector T cells. Fine. Okay, let us come to that once again later. Now, see, so this innate immunity, what is their major function? Their major function means what? They are the first line of defense. So how they identify the microbe? So they identify the microbe with some patterns. Each microbe has some patterns, and these patterns are called as pathogen associated molecular pattern. For example, COVID-19. That COVID-19 virus has a specific pattern on its surface, a protein pattern. That protein pattern only can be sensed by, or that protein pattern will be completely different from the protein pattern which is present in measles or chickenpox virus. So each microbe have their own pathogen associated molecular pattern. Suppose within our body itself, lot of cells will undergo injury. We have learned necrosis, apoptosis. As a result of that also, what should be there? Those should be also recognized, right? So how that is recognized? There we have DAMP. What is that? Damage associated molecular pattern. So this pathogen as well as damage associated molecular patterns are nothing else but a protein patterns which is being recognized by the innate immunity. Fine? Yes. And remember, these pattern recognition receptors, they are present all over the cells. That means they may be present in the mitochondria, they may be present in the cytoplasm, they may be present in the plasma membrane, in the nucleus, everything. Fine? Yes. There are different classes of receptors. Okay? Yes. Number one is what we call as toll-like receptors. Let us see what are these toll-like receptors. These toll-like receptors are first identified in Drosophila melanogaster. Around 10 classes of toll-like receptors are there. I will tell what toll-like receptors have what property they will bind with what component, everything. Okay, they are expressed by what components of the microbes, everything I will express in a very short video, or I will post it in my social media handles. Fine? Yes. Now let us see what is this toll-like receptors going to do. Remember children, this toll-like receptors has two fate. Number one, it is going to activate interferon releasing factors. Number two, it is going to activate nuclear factor kappa B. NFKB means nuclear factor. This is kappa, big K, kappa B. Fine, just remember these things. Why? Because there is a condition called as Hodgkin's lymphoma. We will see in hematology. In Hodgkin's lymphoma, there is activation of nuclear factor kappa B. There is activation of NFKB or nuclear factor kappa B, and that is the main pathogenetic mechanism for Hodgkin's lymphoma. We will see at that time. Fine. So that is about what? Toll-like receptor. The same thing they have written here. It has two fate, that is in NFKB as well as producing interferon regulatory factors. Okay. Now let us see about node-like receptors. So one class of pattern recognition receptors are toll-like receptors. These toll-like receptors, okay, fine, okay. Next is what we call it as the what? Node-like receptors. These node-like receptors are cytosolic receptors. I told these receptors may be present anywhere. So node-like receptors are cytosolic receptors. That means they are present within the cytoplasm. Fine. And this very important. The node-like receptor, okay, the genesis or the mechanism of action of this node-like receptor is similar to that of the pyroptosis. Yes. Here comes the pyroptosis pathway. See, what is this? Whenever there is any pathogenic or bacteria, what will happen? The nod-like receptor protein 3 will be there, which will be sensing it, which will convert the inactive caspases into active caspases, okay, and it will help in formation of what we call it as an inflammasome. I told you we have two pathways of inflammasome formation. One is the canonical pathway and another one is called as a non-canonical pathway. Am I right? And remember this NLRP stands for NLRP3, no, it is an example of leucine rich protein. Okay, yes, this has been asked in a question in GIBMR. Okay, which of the following is a protein which is present within the inflammasome? Answer is NLRP3, which is a NLRP3. That means it is a leucine rich protein. Okay, and this inflammasome will convert the inactive caspase 1 into active caspase. So, and I hope you remember an AIMS question. Okay, an AIMS PG entrance exam question, that is gas dermin. Gas dermin is a protein. That protein cleavage occurs within this step only. Okay, and which will result in the formation of interleukin 1, and this interleukin 1 is responsible for the pyrogenic, that is what, fever, okay, pyrexia and acute inflammation. So this is the pathway, or this is the mechanism of action of nod-like receptors. Fine. And remember this nod-like receptors, sometimes there is a gain of function mutation. Gain of function mutation means what? We have two types of, we have different types of mutation. Out of that, we have something called as gain of function and loss of function mutation. Gain of function mutation means there will be overactivity, overexpressibility of the gene. So, because of the overexpressibility of the N-node receptor, NOD2 receptors, what will happen? There will be auto-inflammatory syndromes. Okay, auto-inflammatory syndrome. That is, without any stimulus itself, the body will induce inflammation. So, the patient will present with intermittent attacks of fever. And these nod-like receptors are implicated in the pathogenesis of 4 major diseases. These 4 major diseases are VVIPs. Okay, these 4 major diseases are VIP. One is type 2 diabetes mellitus. Number 2 is atherosclerosis. Number 3 is Crohn's disease. Crohn's disease, it is VIP. Crohn's disease is one of the type of inflammatory bowel disease. We have two types of inflammatory bowel disease. One is called Crohn's disease, another one is called ulcerative colitis. In Crohn's disease, the pathophysiology is that it is the overactivation of nod-like receptors. Fine? Yes. Okay, that is Crohn's disease, and we have something what we call as the gout. So please remember these 4 diseases. These 4 diseases has been mentioned in your Robbins 10th edition. You should remember this for sure. Okay, you may expect any type of MCQ from this. Okay, once this is done. Okay, once this is done. So I hope you are clear with the pathway. I hope you are clear with the pathway. Yes. Now let us see that other types of receptors. So we have discussed nod-like receptor, tau-like receptor. We have something called the C-type receptors. What is that? C-type lectin receptors. C-type lectin receptor. Remember, C-type lectin receptors are mainly involved in fungal infection. Then we have something called as RIG, that is retinoic acid inducible gene. RIG-like receptor is for viral infection. So they will simply give you a match to the following question. Okay, RIG, nod, TOL, okay, C-type. So what all infections they are going to involve? Fine, yes. We have something called a STING, that is stimulator of interferon genes. Okay, so what is actually, it is nothing else but the STING pathway. Okay, just remember it has been asked in GIBMR once, what is the STING pathway? The STING pathway is responsible for the formation of antiviral cytokines. Again, I told no, RIG is for antiviral, so it detect the viral RNA. Similarly, we have STING pathway, they will produce this antiviral cytokines. Whenever there is a problem, okay, or excessive activation of this pathway, it will cause a condition what we call as interferonopathies. What are they called as? Interferonopathies. Okay, like autoinflammatory syndrome, overactivation. Here we have interferonopathies. Now let us see a very important cell. Natural killer cells. They are a part of innate immunity or acute immunity? Innate immunity. They are what type of cell? They are innate lymphoid cell. What is their specialty? They don't have T cell receptors. Fine. They mainly target against virus infected cells and tumor cells. This point is important. So whenever in the question they have been, which of the following are the cells which are most help in destroying the virus infected cells or tumor cell? Your answer should be first NK cells, natural killer cells. Fine. Yes. They express a receptor for the immunoglobulin. Fine. And this will be able to lyse what? The immunoglobulin coated target cell, and this phenomenon is called the antibody dependent cellular cytotoxicity, ADCC. Just remember this, you will see in detail in immunology and microbiology, and of course in coming session we will see what is ADCC. Remember, not only the natural killer cells exhibit this property. We have macrophages and some other cells which have the phenomenon, which shows the phenomenon of antibody dependent cellular cytotoxicity. Fine? Yes. Why I say antibody dependent? Normally it is the T cell. we have no CD8 cytotoxic T cells. They are only mainly responsible for killing of the cells. Whereas NK cell, by using antibody receptors, they will cause a cell lysis. That is why it is called antibody dependent cellular cytotoxicity. Fine. How this NK cells are active? Remember, the NK cells, they mainly what we call the secrete interferon gamma, interleukin 2, 15. Okay, please go through all this cytokines. Not that much significant. What is the most important concept they will ask here? The most important concept they are going to ask here is how these NK cells are going to, their control of these NK cells. Number one, by inhibitor receptors A, and number B is by activating receptors. We have two types of receptors in NK cells. Activating receptor. So this is the normal cell. You can see in this picture, this is the normal cell. Suppose if the inhibitory receptor binds with the MHC class 1, we have major histocompatibility complex. We have class 1, class 2, class 3 and all. In class 1, suppose if the MHC 1 binds with the inhibitory receptor, since it is inhibitory, NK cell won't get activated, no cell killing, simple. Suppose if the activating receptor is there, so what is going to determine whether the activating receptor should be activated or the inhibitory receptor should be activated? So there should be some reason, no? See, in first thing I told that the inhibitory receptor is there because the cell will express, a normal cell will express MHC class 1. A normal cell express MHC class 1. When a cell has MHC class 1, it will always bind with the inhibitory receptor of the NK cell. That is why there is no cellular killing in normal condition. Whereas in case of viral infection, I told no viral infection tumor, in that there will be inhibition. The virus is going to inhibit this MHC class 1. So this MHC class 1 can't be expressed. So the MHC class 1 can't be expressed means, of course, the inhibitory receptor cannot bind. Because I told inhibitory receptors will always bind with MHC class 1. Since in virus infected cell, the MHC class 1 is not being expressed, the inhibitory receptor has no role here, then who will bind? Of course, the activating ligand will bind because it is a MHC cells, MHC1 is not expressed. The inhibitory receptors has no role here. Activating receptors activated. NK cell activation, killing of cells. Please, very, very important, very basic question. Very basic question, straight away one-liner MCQs from this you will get. This concept is very important for understanding of, because I am not going to skip each and every, whatever may you in Robbins, I am going to tell each and every point, because each and every point is very important for understanding all the autoimmune disorders, hypersensitivity reaction, even in neoplasia, everything is important. So that is the mechanism of NK cells. So that finishes innate immunity. Now let us see adaptive immunity. I told we have lymphocytes, B lymphocytes and T lymphocytes. Adaptive immunity, again we have humoral and cell-mediated immunity. Humoral immunity is B lymphocyte, bone marrow derived, they produce antibodies. T, cellular immunity or cell-mediated immunity is thymus derived lymphocytes. Fine? Yes. And remember, we have different types of lymphocytes. Okay, lymphocytes, they are formed from the lymphoid lineage from the hematopoietic stem cells. So, we have something what we call it as naive cells. What are naive cells? Naive cells are those which are produced. They are that means they are immunologically inexperienced. Okay, I have written unexperienced immunologically. Okay, that means they have not encountered any antigen. They are just produced. Like how a newborn baby, am I right? When the baby comes immediately from the mother's womb, he is new to this world. Am I right? Similarly, these lymphocytes that they have been produced just from the bone marrow, say they will be naive. They don't know, okay, what's happening around. They have not exposed any strangers or any antigens. So they are called as naive cells. Fine. These naive cells, then they later start activating, they get encounter the antigens, and finally they differentiate into memory cells, effector cells. We have different types, we will see that. Remember, yes, the lymphocytes specific for a large number of antigen exist before exposure to antigen. Am I right? We have lot of lymphocytes. See, I told you naive cells. So, suppose I am drawing here, this is one lymphocyte. I am drawing dot dot dot. These are various antigens. Am I right? Fine. So, before these lymphocytes are exposed, before exposure. Am I right? Yes. Okay, we have lot of lymphocytes. So we have a lot of lymphocytes and lot of antigen. But once an antigen comes to bind with the lymphocyte, each lymphocyte will bind only with a specific antigen. For example, I am telling, let it make it more clear. We have A antigen, B antigen, C antigen, D antigen. Initially, each of this lymphocyte can bind to any of the antigen. Fine? Yes, they are immature. But once they become mature, what will happen? This 1, 2, 3. Okay, so this one lymphocyte, they may specifically bind only to D antigen. 2 lymphocyte will bind specifically to C antigen. 3 lymphocyte maybe specifically binds to A antigen. So that is specificity. That is called the clonal selection. That is called a clonal selection. That is called what? Clonal selection. Fine? Yes. Clonal selection is a very big topic. You will see in depth in micro. Now let us see the functions of each of the T lymphocytes. Okay, each of the lymphocytes. We have T lymphocytes. I told you, we have T lymphocytes. Then we have B lymphocytes. Okay, all those I am going to tell in the next few minutes. Okay. Now let us see what are the functions. B lymphocytes, they mainly help in neutralization of microbe phagocytosis. We have different classes of T lymphocytes. T helper lymphocytes, they mainly help in activation of macrophages, inflammation and the proliferation of lymphocyte. Cytotoxic T cells, killing like our NK cell. But remember here, this express T cell receptor. Okay, it is not ADCC. It's not antibody dependent cell immunity. Antibody dependent cell immunity is specific to NK cells. Then regulatory lymphocytes, which will suppress the immune response, which will prevent the exaggeration of immune response. Fine. So T lymphocytes, I have told you all these things. Fine, they develop from the thymus. Fine, and see, they develop in the thymus, but they arise from the hematopoietic stem cell. So each T lymphocyte, I am going to draw the picture here. See, this is a T lymphocyte. I am drawing here. This is a T cell. This T cell will express something what we call it as T cell receptor. This T cell receptor have alpha domain and beta domain. This alpha and beta domain will bind with the alpha and beta domain of dendritic cells. What is dendritic cells? Dendritic cells are nothing else but what we call it as antigen presenting cells. Dendritic cells are nothing else but what we call it as antigen presenting cells. Okay, so these dendritic cells, okay, they will express class 1 or class 2 MHC. Class 1 or class 2 NMC. I have drawn a CD4 T cell. Remember, CD4 T cell always express class 1 MHC, class 2 MHC. Okay, whereas CD8 T cells, they always express class 1 MHC. Okay, how to remember this? 4 into 2 is 8, 1 into 8 is 8. That means CD4, that means I am writing here, CD4 cells express MHC2. CD8 cells express MHC1. So since I have drawn a CD4 T cell, it has a T cell receptor having alpha beta domain. This alpha and beta domain will bind with alpha and beta domain of the class 2 MHC, which is present on the dendritic cell. Because dendritic cells are antigen presenting cell, they are the cells which are going to present the antigen via MHC through the T cells. Fine? Yes. Whereas this classical presentation won't be seen in natural killer cell. In NK cell, there is no T cell receptor, no MHC, nothing. Okay, it don't depends on MHC and all. It depends only on the immunoglobulin receptor, IG receptors. Fine, that is why it binds with the FC receptor. Okay, we have FC and FAB regime, FAB receptors. Okay, 2 types of receptors there in immunoglobulins. So out of that, they use the FC receptor of the immunoglobulin, and they using that they will bind with that of the using that they will bind, and they will depend mediate cell killing via antibody dependent cellular cytotox. That is called, it is ADCC. Here there is no antibodies because here MHCs are present. Fine? Yes. So you can see in this picture also. See, this is the CD4 T cell. Fine. They express T cell receptor which is having alpha and beta domain. Okay, please neglect about these things, not needed for you. Fine. And this will bind with the alpha and beta regimen which is present in the antigen presenting cells of the class 2 MHC molecules. Am I right? Yes. Okay. So this is how a T cell receptor will bind with the MHC as well as on what? Antigen presenting cells. So this is a response from T cells to MHCs and APC. So when I am doing a favor to you, normally the rule of the nature is I will also expect you to do some help in return. Yes, that is the normal human. Now, normal human psychology. Am I right? Similarly, since T cell receptor is sending some stimulus and it is binding with APC, the T cell receptor will also what? Expect the APCs to do something in favor. What is the APCs going to do something in favor? See, for T cells to respond, okay, they are so what? They are head-weighted people, okay. Yes, some ego problems T cell receptors have. So what they will do? No, I have bind, no. He should also send me signals in return. Then only I will start the reaction. Yes. So after this T cell receptor binding with APC and MHC takes place, the APCs will send some signals. Okay. The APCs will send some signals via two important molecules. That is CD80-86 and CD28. We will see that in detail again in autoimmunity and all. Okay. So the CD80, CD86 of the APC will send signals to CD28 of the T cells. You should remember number 2 points. CD80 and 86 is in APCs. T cells are present in, CD28 are present in T cells. I hope this is very clear for you. These are things they have mentioned here also. I am telling again, CD4, 4 into 2 is 8. That means CD4 cell always bind with the MHC2. 1 into 8 is 8. That means what? MHC1 always bind with the CD8, or in return CD8 binds with the MHC1. Okay, please remember this. T cell receptor have alpha and beta domain, that will be bind with the alpha and beta domain present in class 2 MHC if it is a CD4 cell, or class 1 MHC if it is a CD8 or cytotoxic T cell. And I told no, ego problems are there. So if the T cell has to start the immunity, what the T cell will expect? That APC should respond. How the APC respond? Via CD80-86, it will send signals to CD28, which is present on the T cell. CD28 is on T cell, CD80-86 is present on APCs. I am telling repeatedly again and again because it is very important. Then we have B lymphocytes. These B lymphocytes constitute around 10 to 20% in the peripheral blood. It also has some B cell receptors. They will transform to form what we call the plasma cells. This plasma cells secrete antibody. We have different classes of antibodies which you will read in biochemistry. Fine? Yes. Here we have two types of See, here also we have activation. Okay, activation. Like how they are binding, no? The T cell receptors bind with MHC 1, 2. Here we don't have any MHCs and all. Okay, here some important receptors are there. Okay, number one is that this B cell receptor will bind with the B cell receptor will bind with CD21 or CR2, type 2 complement receptor, or CD21. So that is one way. The second way is CD40, CD21 or CR2, or next one is what we call the CD40. These are the two receptors which we will use for binding, which will further activate the immune reaction. Not that much significant as of now, but CR2 is very, very important because it is used by the Epstein-Barr virus. Epstein-Barr virus also express CR2 or CD21 receptors, and these receptors are similar to the receptors present in the B cells. So using that, they will invade our immune system. They will cause severe infection. Okay, they will have severe infection. Viruses are very smarter. Okay, what receptors we are having in our body, they are evolving. The viruses are evolving and they are developing those receptors. And using that receptor, they will get protection from our own immune system. Fine. We have third and last, dendritic cells. They are the most classical antigen presenting cells. They are going to present the antigen to the T lymphocytes. Where they are located? They are located in epithelia. Some immature dendritic cells are there which are present. Immature dendritic cells are present in the epidermis of the skin. Very important, I am telling it again. The dendritic cells which are present only in the dermis we call as Langerhans cell. If it is present somewhere else, please don't use that name. It expresses many receptors, fine, and they present antigens to the T cells. They have very high level of MHC, so they will immediately activate the T cells. Okay, yes. So these are the normal type of dendritic cell. We have a specialized type of dendritic cell called as follicular dendritic cells, short form FDC. These follicular dendritic cells, remember, as the name indicates, follicular, they are present in the lymphoid follicles. Where are these lymphoid follicles present? In the spleen and lymph node. Okay, only there these type of dendritic cells are present. Fine. Yes. Okay. And they have, these type of dendritic cells have receptors for immunoglobulins. Okay, and they play important role in humoral immune response. This is the difference. The difference between follicular dendritic cells and the normal dendritic cell. Normal dendritic cell, they express the antigen towards what? MHCs and all those. So what is that immunity? Adaptive immunity. Sorry, cell-mediated immunity. Whereas the follicular dendritic cells, which are present within the germinal centers of the lymphoid follicles in spleen and lymph node, they have what? FC receptors for immunoglobulin, like our NK cells. NK cell use also this what FC receptor of IgG, and they causes antibody-dependent cellular cytotoxicity. Similarly, here also the follicular dendritic cells also bear these FC receptors for immunoglobulin, and they play a role in important what? Humoral developing. They are going to start the humoral immune response. This has been a question asked in JIPMER. Okay, all of the following statements are true except, or true with regard to follicular dendritic cell except. Okay, what is the answer? You know, the answer was that the option they have given, that is follicular dendritic cells evoke cell-mediated immunity. No, that is wrong. Follicular dendritic cells evoke humoral immune response. Again, the same concept we will learn in detail in your microbiology. Then macrophages, already you know the functions of macrophages. Then we have two types of lymphoid tissue. One is primary lymphoid organ, where the cells are produced. Am I right? What are they? Bone marrow and the thymus. Fine? Yes. And secondary lymphoid organ, we have lymph node, spleen, malt, mucosa associated lymphoid tissue, galt, okay, bronchial associated lymphoid tissue, all those things. Okay, and rest of the primary lymphoid organ, secondary lymphoid organ, their functions, everything we will see in the next session. So today in whole, we have discussed about right from the very basics of immune system. You can't afford to miss even a single point. Each point is important because a lot of complicated stuffs you have to read in the chapter immune system. At that time, each of this point only will help you to build up your concept. Of course, you know, if you read this in a single day, you can't understand, you can't remember the points. Revise it frequently. Okay, and soon we will see you in the very next session. Okay, in the part 2 of immune system. Of course, immune system is a very extensive chapter. Of course, okay, I don't know how much parts I will be making. Of course, all these are 30 minutes videos, short videos only. Okay, because you will get saturated more than 30 minutes in immune system neoplasia chapters. So I am making just 30 minutes video. Fine. Please take notes on this. Please mark these points on Robbins. Highlight those points. And why I am highlighting these points? Because these are the things you should revise before your internals or your university exam, because at that time you can't sit and read what all the stuff. So take separate notes from this Robbins. Fine, that only will help. You can't go through the lines because you will have just one exam one day. Okay, mostly you won't have gap between two papers, paper 1 and paper 2, and of course you have to sit you have 2 volumes around 1500 pages you have to finish within 2 days, and that too reading Robbins just before the exam. It is very hectic. So please maintain proper notes and please highlight. Please don't use multiple colors in highlighting. Again, it is very difficult for you to revise. Use a single color, okay, and try to underline the highlight. Only highlight means please highlight only the important point. Don't highlight each and everything. Then what is the difference? Okay, yes. So please do that. Then only you can revise and keep all these points in your memory. Thank you.